that these germs, these microorganisms, were responsible for these diseases. So scientists set out to prove it. Are these microorganisms causing disease? Can I match a specific microorganism? And again, they've been working with them and they can see they're different. Can I match a specific microorganism to a specific disease? So Pasteur started working on it, but many other scientists did as well. And there was one scientist called Robert Koch. He was German. He lived in a, this is the PowerPoint about spoiling people and that germ theory. He lived in an agricultural area, and he was familiar with a disease that affected cattle called anthrax. Now, nowadays, when you think of anthrax, you think of bioterrorism. But back then, this was a very common disease that would affect herds of cattle, and it could be fatal. It could be very bad. It was a skin disease, caused a lot of these bad lesions on these animals. All right, so he was looking at this disease and trying to figure out if there was a microorganism that was responsible for this disease. So just like Pasteur and the scientific method, he went through a very step-by-step process to determine if he could find a microorganism that caused this disease. Now, pardon my little whiteboard here, but it seems to be the best way I can get information across in a video. Also, remember, I can't draw. Couldn't draw in the classroom, can't draw here either, but this is my cow. So we're looking at a cow. This particular cow has anthrax, which is a skin disease. So he has some skin lesions on him. And Robert Koch took scrapings from these lesions to see if there were microorganisms there. And so he did that, and he grew them outside the lab. And at this point in time, they didn't have plates, agar plates like we do now, and that we grow things on in lab. And so he actually spread it on a slice of potato. And the potato served as a food source for the microorganisms. And sure enough, he got some growth. And he observed that there were different microorganisms that were present in those skin lesions. Quite a few, just like Pasteur, there were different types, and so there are quite a few. And so his question is, does one of these microorganisms cause anthrax? So how do you prove it? So he took these microorganisms and he introduced them into a new host. So he took the purple, okay, so these now, a new host, these look very similar to my cows, but he actually switched to mice because they also can get anthrax. So again, my mice and my cows look the same. Doesn't really matter. So he took a new host and he took this microorganism. He put a cut in the skin and rubbed that microorganism, just that one, into these different animals. So we had the triangle, the blue triangle, the red circle, the green X, and each one of these got one microorganism. Again, one variable determining if this animal gets anthrax or not. So, observations. What happened? Well, this animal didn't get sick. That's a smiley face. This animal, oh, that looks so bad. And this animal didn't get sick. So when he did the blue triangle microorganism, whichever one that was, it was healthy. It was fine. So there was no anthrax involved. So his conclusion was that this blue triangle microorganism, whatever that was, does not cause anthrax. Okay, fair enough. Even when you get a negative result, or in this case, you know, a result not what you're wanting, you get some information from that. So he knew that the blue triangle microorganism did not cause anthrax. Now, this animal with the red one did get sick. However, this disease was not anthrax, something completely different. It caused some signs and symptoms, but it wasn't anthrax. So he can conclude at this point that the red circle bacteria does not cause anthrax. All right, what about the last one? This one does get sick. This one does cause anthrax. And so you may think that the conclusion from this one is that the green X bacteria does cause anthrax. And that's possible, but how do you know that this cow didn't get, or this mouse just didn't get anthrax because sometimes animals get anthrax? Was it a direct result from what you did? that you gave this animal, this host, the microorganism, and this organism got sick, is this a direct result from what you did? Well, you need to kind of rule that out, that he didn't acquire anthrax in other ways. So how do you do that? And this was the last step that Koch did, and he took this animal, and remember it's got anthrax, so it's got these skin lesions on it like before. He took scrapings from these skin lesions and then put it on a potato, on that plate, so that he could grow it in the lab. He wanted to know, was this microorganism present? And yes, when he did that, he was able to re-isolate and find that microorganism as well. The other important thing that you need to know is that when you take samples from a healthy host and grow that, you may get other microorganisms, but you cannot find the organism that you think gives you anthrax. So that's also something that was helpful and able to rule this out. And again, scientific method, making a good experiment. This was a fully thought out experiment. And he determined that this green X, which is now known as Bacillus anthracis, is the cause of anthrax. All right, so he was the first one. He was the first one to match a microorganism to the disease, so he gets credit for that, even though Pasteur put forth the germ theory. His rules and his steps, he wrote them up, and we now refer to these as Koch's postulates. So when you go through these steps, there's a certain amount of things that have to happen before you can conclude that the microorganism you found is the cause of the disease, or anything is the cause of what you're looking for. Sometimes things happen and they're correlations, but not causes. And we end up working with that closely today. A lot of times you find things that happen, but it may not be the cause. So you're always looking to identify the cause and not just something that's, that happens, just because. All right, Koch's postulates, what do they say? First step, the suspected agent must be found in every case of the disease and absent from healthy hosts. So every time you sample the agent, a sick host, you must find that microorganism. Every time you sample a healthy host, it should be absent. You should not find that microorganism. So that's number one. Step number two, you have to be able to isolate it and grow it outside the host. Remember, he did it on slices of potato. Three, when you take that microorganism and you introduce it into, a healthy host, it must cause the same disease. So must cause in this case, anthrax when it's introduced into a healthy experimental host. Then that last step, you have to be able to find that microorganism in that diseased host. All right, so this was a very scientific way of determining a cause from that. We still use this today when we're looking at it. There are some drawbacks to this. So take a minute and think, go through these steps, and if he had been looking at a different disease, would he be as successful? If he had been looking at flu or chicken pox, would he have been as successful? If this disease was not caused, just think about it for a second. Okay, so things to think about. If he had been looking at a disease that was a virus, he would not be able to grow that outside the host at that point in time. So he would never be able to do that. So again, a lot of work and some luck allowed him to be the first person to match this up. But if he had been looking at a viral disease at this time period, he wouldn't have been successful. If he had been looking at a disease that had multiple causes, he wouldn't have been as successful. If he was looking at a disease that was not infectious, genetic diseases, something like that, he would not have been, or environmental causes, he would not have been as successful. All right, so again, we use Koch's postulates. It'll come back again in this course, so make sure you learn them. But there are some drawbacks, especially to the diseases we're looking in our modern day age. Okay, so causes of disease. We've got one. We finally found one. This led to a whole new branch of science called etiology. Etiology is the study of what causes disease. Robert Koch was also very instrumental in some other things. He did a lot of work with microorganisms right at the beginning. He was the one that took a metal loop and was able to use that metal loop to transfer bacteria. We still do that now. You'll see that in your simulations. He was the first person to do some of the aseptic techniques that we use in lab to keep down contamination. His associate was named Robert Petrie. And again, the potatoes got old, they would, rot. And so they came up with the agar that we use today in various forms, where you took agar, which is that form of algae, and were able to use that, adding nutrients to it as a source where the microorganisms wouldn't degrade the agar plates. So they did that. So they did a lot of things, the staining techniques, some simple staining techniques, a lot of techniques that developed in that laboratory that we still use today. So he was very important and instrumental in that.